And we are so glad to have Dr. Marco Rolandi as our next keynote speaker for today. Dr. Rolandi is the Jack Baskin Engineering Chair and Professor in the Department of Electrical and Computer Engineering at the University of California, Santa Cruz. He is also co-founder and chief scientist advisor of Cruise Foam that makes sustainable packaging solutions. Dr. Rolandi received his PhD in applied physics from Stanford University and a lead certificate from Stanford Graduate School of Business. His research focuses on bioelectronics systems and devices, biological control in regenerative medicine and synthetic biology, and their translational applications. His work has been highlighted in the New York Times, Washington Post, the BBC, New Scientist, IEEE Spectrum, and several others. He currently leads one of the teams performing on the DARPA BTO BTRA programs to accelerate wound regeneration using bioelectronics. Welcome, Dr. Rolandi. Thank you.
Okay. Thank you so much for the introduction and for the invitation to speak here. I would have loved to be there in person. Unfortunately, my travel got a little bit complicated the last couple of weeks, and so I'm back in California. I know it's getting late there in Minnesota, so I'll try to keep it brief, and I'd love to visit in another point. It sounds like a really exciting meeting. So today I'll discuss the work of a fairly large team, actually, and I'm glad the prior speaker, mentioned that, these days innovation in medicine, I would say pretty much innovation in anything, usually is made by large interdisciplinary teams. And I've been fortunate enough in the past 6 years to be able to lead a team, or to work rather with a team of many gifted PIs and graduate students and researchers in a project funded by DARPA and ARPA-H. And the project aimed at closing wounds in 50% of the time. And the idea was to develop a device that would let us do so. Now, before I jump into the project, perhaps I can tell you a little bit how we got to designing this device and the sort of like the principles behind it. So I'm going to start from a very basic principle, which is homeostasis. Homeostasis is dynamic equilibrium. And, one could argue that pretty much homeostasis is the center of life. Very classic example is how do we maintain our temperature for optimal, as warm-bodied mammals, our temperature needs to stay constant within a range, because otherwise our biochemical reactions don't work very well, and a lot of bad things happen if our temperature becomes too low or too high. So the way it works is that we have a set of sensors fundamentally that are able to detect our internal temperature. If our temperature becomes too high, what happens is we typically start sweating and a few other things. And then the sweat dries off our skin, latent heat removes heat from our skin, our body temperature drops. On the other hand, if we are too hot, we are too cold, what happens is that our sweat gland stops secreting fluid and we start shivering. And by shivering, then we sort of move, and by moving, we rebuild heat. And this is a very classic control system problem, in that it's what people will refer to as a closed loop control or feedback control in engineering. Fundamentally, almost every system in our body functions like that, whether it's a memory voltage of the cell, whether it's the pressure inside the cell, whether it's the blood, the glucose level, and so on and so forth. And the technological analogs, for example, are self-driving cars. We started looking into this concept a few years back, and the idea was that one of the insights was that most of the time when something fails in our bodies, it has to do with this homeostatic loop not working. For example, for diabetes, then what happens is that the pancreas doesn't secrete insulin anymore, and so the blood glucose level goes all over the place. And so the idea was that, well, instead of using either replacement insulin or drugs for other type of homeostatic problems, is there a way to insert ourselves into the loop actually fix that. And this is a concept that we didn't invent. There's a lot of work in electroceuticals in general that follow the same idea. And we started from the very foundation. So this work was done with Marcela Gomez, who is a control system specialist in applied mathematics here at UC Santa Cruz, and Michael Levin, who is an evolutionary biologist at Tufts, one of the experts in bioelectricity. And we started with very fundamental concept, which is the membrane voltage. And the idea was that, well, if we can control membrane voltage that influences a lot of the biochemical reactions, then we can then expand this to a more complex system. So what we did is as follows. This is a cartoon of the work. And the pink thing down there is a cell. And we have means of measuring the membrane voltage with the fluorescent reporter or arc light. And this reporter, the fluorescence intensity goes up when the membrane voltage is high as in hyperpolarized, so more negative, and the fluorescence goes down when the Vmem is low, as in hyperpolarized, so in a way it's actually less negative. What we do here, we record it with a fluorescent microscope, and we compare that with the target value. The idea behind this was that depending on their membrane voltage, some cells are more or less proliferative, and we wanted to see whether we could control cell proliferation of stem cells, and also whether we could control their fate. Now, this target value is inserted into a neural network, which compares the target value with the arc core value, and then the neural network decides whether we want to actuate or not, meaning that if the target value and the actual value are the same, we do nothing. If they're not the same, we try to affect the surrounds of the cell in a way that we can actually modify the membrane voltage. And the way we do so is with bioelectronic actuation. And the bioelectronic actuation looks as follows. Fundamentally, what we do, we use electronic signals, and the power of that is that we can interface this with a microcontroller, a computer, we can send signals through the internet, so on and so forth. And we use this signal to deliver biochemicals and or ions. And we do so with a concept called electrophoresis, in that we have a voltage, and the voltage is able to drive through a solution charged molecules. In this case, we use a simple proton, and what that does, it changes the pH of the extracellular fluid, and affects the channel membrane by the cells, and in turn affects the membrane voltage. So when we actuate, the positive voltage reduces the pH, so on and so forth. And after actuation, we basically see what are the effects of actuation on the cell, whether the MEM has gone up or down, and then we behave accordingly because the system learns, and then we can change the target value. Now, this was a small seed from DARPA, and what they wanted to see is whether this was possible. And when we developed this, and then DARPA built a bigger program specifically on wound healing. Okay. Now, this wound healing is very complex, and one of the common pictures about wound healing, despite the fact it's quite simplified, is that wound healing occurs in 4 phases. And the idea behind it is that there's the first phase is when the blood clots, then there's the inflammatory phase where basically everything gets cleaned up from the macrophages and one inflammatory macrophages. And then there's a proliferative phase in which the M2 reparatory macrophages are starting to rebuild tissue, and then there is remodeling maturation. And what people have observed is that when wounds don't heal appropriately or as fast as we would like, something happens between these stages. Usually there are some issues where either inflammation lasts too long or doesn't last long enough, proliferation doesn't occur. And specifically, when it comes to chronic wounds, in that wounds that don't heal at all, such as, for example, diabetic ulcers, where people thought originally that rather than progressing through these stages, such as shown on the top left there, people thought that basically the wound never left the inflammation. So the wound kept staying inflamed in family place, so it never started proliferating. Now, as part of this work, we actually looked at the data from gene expression from many chronic wounds because we wanted to understand that we needed to intervene on other wounds as well. And Professor Gomez and Dr. Isserov, who is a physician on the team, what they found out is that the gene expression data points to the fact that chronic wounds don't actually never leave the inflammatory phase. But what happens, they actually oscillate between inflammation and proliferation. So they get into this vicious cycle in which the inflammation destroys the tissue, goes into a proliferation, rebuilds it, and then at one point, for some kind of reason, instead of moving into remodeling, it goes back into inflammation, so on and so forth, and therefore the wound never heals. Now, what we came up with this question, and the question is, in these situations, or even in an acute wound that tends to heal, but maybe doesn't heal as fast as we would like, are we able to shift with external means the wound into an optimal homeostasis in this dynamic equilibrium in this many processes occur, and so that the wound heals faster? Now, this was the goal of the project that was funded by DARPA. Started originally about 5 years ago, actually in 2020, just when COVID hit, actually. And the idea was basically to take a normal wound, find a way to predict the probability of all these stages, and that's what's shown here on the left, during the y-axis, that the probability of the stages on the x-axis is actually days, and see whether we can shrink time fundamentally, right? Can we make all of these things go twice as fast? And the principle behind this was that we were going to put together 3 types of technologies. The first technology was the delivery of therapy, which we did fundamentally using the same principle as I shown before, using electrophoretic delivery. And instead of just delivering H+, we deliver electric field and other small biochemicals. Now, in order to decide what therapy we wanted to deliver, we then used sensors. And these sensors are designed to pick up data from the wound, compare it with training data from an algorithm, and decide whether the wound looks like a wound at day 2 or a wound at day 3. And for example, if you are at day 3, we really want our wound to look more like a wound at day 5, meaning that the wound is healing faster. And we're doing so with an ML algorithm that helps us figure out what's going on. Now, the idea was, the first thing we needed to do, as I said, is figure out, like, what is actually the wound stage? If you sort of like shrink time, if you will, you really need to understand what's the actual wound time versus the time outside the wound. And a wound that heals faster might look like a wound that healed at time, at day 10, even if it's day 3, right? And the other aspects, which is really important, is like, when do we intervene? Is it better to intervene at the very beginning during inflammation? Is it better to intervene during proliferation? Is it better to not intervene at all? And ultimately, how do we intervene? In that, like, what kind of treatment is ideal for that kind of scenario? And so these are the boxes represent the 3 parts of the algorithm that really drive this work. And in order to set this up, it was actually quite a big effort, because we started developing the sensors, the actuators, but we also needed a lot of in vitro data and validation for both the timekeeper and the treatment control and whatnot, because sort of like testing the data all in vivo would be too complicated. And so what I'm sort of showing here in the next few slides is actually the culmination of these 5 years of work of all of our collaborators and whatnot that resulted in many smaller papers in which we have data for in vitro, we have data for the fluctuation development, the algorithm and whatnot. But the work that I'm presenting right now is what kind of like brings everything together, and it was just recently published in PJ Bioengineering. So this is how it looks like. We have, after many studies, we decided that the best way to actually detect the wound was using a camera. And the idea behind it is that when a physician needs to figure out if a wound needs to be treated or not and how, the first thing a physician does is looks at the wound optically, and then decides whether more analysis is needed, whether more tests such as swab, for example, for bacterial infection. And then the physician uses their experience fundamentally to understand what treatment is needed. And so basically the device itself, the camera represents the eye of the physician, and the boxes are the algorithm. The camera represents, if you will, the experience and the brain of the physician. And then like the actuation represents, for example, it will be a topical treatment. Now, this is not designed to sort of like replace physicians, and in line with what the speaker sort of presented before, we need more physicians. The ratios are, so it's really designed to augment their reach or make them more efficient. And in order to do so, there's also a remote monitoring aspects where the physician can come in the loop if needed, and monitor the wound remotely in case the wound is not healing appropriately or the device is not able to diagnose appropriately. So then the physician can decide whether they need to intervene with the treatment manually, or maybe they need to bring the patients back to the office for a visit. So how does this look like? So this is how the alternate device looked like. This was designed for large animal prototyping and studies. It's not pretty yet. You can see the first one was really definitely not pretty, and then it got better and better. We used an ostomy skin barrier as a support because we didn't have the resources and we wanted to develop too many more things. And that's how DARPA works. You have to solve a very big problem, but you really need to try to use as much of existing technology as possible. The form factor clearly needs improvement because as of right now, it's not particularly comfortable to work on. And we actually have an improved device that is flexible and so on and so forth that is being published right now. But it did serve its function pretty well. So the inside of the device looks as follows. This is the exploded CAD. What you see here is in transparency. I don't know if you can see my pointer is actually the reservoirs that contain all the different drugs and the electronic actuators. There is a small motherboard that has a memory, there's a wireless transmission, so on and so forth. There's a bank of LEDs designed to illuminate the wound. There's a lens, and a camera, and like an enclosure, so on and so forth. All the transparent stuff is silicone, so it's designed to be somewhat flexible to conform for the wound itself. And this is actually the cleaner version. There's different sizes. This is designed for a full thickness wound of about 1 inch, and it's designed for a porcine model. So there's kind of like a little indenter to make sure that the device and the treatment touch the wound appropriately. And the cable is an HDMI cable that connects to an external battery that is wear by the animal itself. For now, we're working on reducing power to minimize the weight load for the animal. If we take a picture from the bottom, this is how it looks like. There's a camera with all the board and stuff, and these yellow things are the LEDs. And one of the things we had to do, we had to spend a lot of time making sure that the PMS or the silicone was transparent and clear. Then we can look at the image nicely. This is an image with what you see here are the wires that connect the wound to the electronics. These divots here are the reservoirs with the drug, and this is like a banana slug, which is the mascot of UCSC, and you actually find it on campus a lot. This is a wound image. It doesn't look great, but the nice thing is that because you're training the algorithm on all the same images, and we basically have physicians look at the images of the wounds without the device on and tell us whether the images are infected or inflamed or not. And then we validate this with genomic data. What that means is that the algorithm is trained on images that are not perfect, and therefore it can actually predict the wound stage with images that are not perfect. So what does the algorithm do? What we needed to do first is create a linear model that basically would predict wound trajectories. And what you see here on the left is the percent healed as a function of time. And those boxes, those color boxes represent the percent or the probability of the wound being in a specific phase as a function of time. Red being hemostasis, then like inflammation, proliferation, and maturation. And the yellow is the trajectory of the wound that is not healing as fast as we would like. And what we try to do is to move the wound into a trajectory that is much faster, such as the one shown in green. In order to do so, the way this works is that the model takes an image of the wound, puts into an algorithm called deep matter mapper. What this does, it actually creates an image, a digital twin image fundamentally of the wound, how the wound should look like in an accelerated trajectory, feeds it back into another algorithm that compares with the wound and decides whether the two wounds are the same or not. And by doing so, it will then decide whether we want to treat it. And this is how it looks like. So there's the actual image itself. The deep mapper decodes it, figures out the wound stages. The wound stages are then placed into this linear model, and they tell us whether the wound is in the right trajectory or not. And then the linear model spits out or creates a target stage, and then the encoder encodes an image of the target stage into an artificial image of the wound, and actual and target are compared. Now, when these two are compared, if they're the same, we do nothing. If they're different, then we have two options for treatment. One is an electric field, and the other one is actually the drug fluoxetine, otherwise known as Prozac. Turns out Professor Issaroff, a dermatologist at UC Davis, works with us. I've done a lot of topical studies on Prozac and shown that it really helps the wound close faster by reducing inflammation. We think it has something to do with innervation and sort of connectivity between the wound and transmission, nerve transmission between the wound, because fluoxetine is a serotonin reuptake inhibitor. However, we haven't gone into that as much yet. So this is how the actuators look like. What you see is the motherboard with the PDMS. These are these are actually food coloring. And the reason for the food coloring is so we can detect which ones have basically an ionic cocktail that is designed to make it conductive, so we can deliver electric field to the wound. The electric field, what it does, it basically drives the cells to assemble the cells in a better way in the wound. When one has a wound, there's already an innate native electric field. And people have found out, Dr. Minzao and our team, that if you augment the electric field in the wound actually closes faster. Now, so the blue represents the channels, if you will, that deliver electric field, and the red represent the channel that deliver fluoxetine. So this is a cross section. There's a motherboard, a PDMS, the channels. What you see here is actually what's transparent. What you don't see, it's a hydrogel that acts as a filter. We need the filter to make sure that we're only delivering the molecules that we want rather than everything else that's in solution. And the filter, what it does, it filters through is a cationic hydrogel. And so it lets filter the positive charges through, and it doesn't let the negative charges go through. So when we want to apply electric field, we activate the channels that have these ions in there. And when we apply fluoxetine, we activate the channels that have fluoxetine in there, representing in blue and yellow respectively. We did a little bit of simulations to figure out, to making sure that what we're doing actually made sense. What you see on the left is a COMSOL simulation that takes the current measured during the treatment and evaluates electric field apply. And the wound electric field is about 100 millivolt per millimeter. And what that means is that we can double it to make it better. And on the right, we actually can predict the dose of fluoxetine delivered by basically measuring the current and figuring out how many molecules of fluoxetine are delivered for amount of current. And so we can get a pretty good sense of the dose that we deliver for each wound. Now, this is not one of the things that we do before in vivo test. We actually found out that the chicken breasts are a very good model for testing devices. And they're fairly easy to find, and then we can just go to the grocery store and we can fairly test it fairly easily in an engineering lab. And it was very important to do a lot of work to testing out all of this stuff before deployment, making sure the contacts are good, making sure that the transmission is good, so on and so forth. And we call it the impullum test. So we basically went from in vitro to impullum to in vivo as an intermediate step to make sure this worked. We designed the experiment at UC Davis. It uses a swine. And the way the experiment works is that we treat the wound for the first 7 days, and then we have standard of care. The reason why we only treat the wound for the first 7 days are twofold. The first one, that we found out that treating the wound early on has the biggest effect. The second one, it was practical, is that because we have to change the device every 3 days or so, the typical time it takes to clean up the, for changing the dressing as well, if we were to treat the wound for a time 22 day, we just had, for a simple animal test, we just had to make too many devices. These are handmade in the laboratory, and we just couldn't make enough devices that were reliable to actually run an experiment this long. And so basically running it too long would increase the risk of failure and getting no data at all. So this is how it looks like in the device itself. And you see these are protected, and then this one has a jacket. It can move around, roam around freely, for better or worse. The worst part is they're often they roll in the mud and then they mess everything up. So that was a little bit complicated, but nonetheless, the system did what it was supposed to do. This is a plot of the probability of the wound stages, so hemostasis, inflammation, proliferation, maturation, is a function of time for the first 7 days from devices based on an in vivo experiment. Now, what you see here is the background is actually a treatment, which is blue for electric field and pinkish for Prozac rather. And the reason why it switches, what we found out in preliminary studies is that the electric field has the biggest effect to reduce inflammation. And once the inflammation is going into being less and less, then we can switch to fluoxetine. And so the system is designed to do the switching at about 40% inflammation. So once the inflammatory stage reaches its peak around 40% and starts dropping, then we switch the treatment on the same device, because this is a combination device. And you can see how this looks like from our measurements, from our data, where we have plots as a function of time, and on the left is the electric field strength, and on the right is the rate of fluoxetine delivery for each device. So there are 8 points of delivery on the wound. The one labeled odd are electric field, and the one labeled even are fluoxetine. And you see the electric field gets delivered at the beginning, then we stop, and then we transfer to fluoxetine. The fluoxetine delivery happens about 6 hours a day until we reach a maximum dose that we also found out was the optimal dose in other studies. And then the system stops, then it waits until the day is over, and it starts again when needed. So, well, this is a plot of the data when we put this in the linear model, and the green dots are the control wounds, which are ones that have the device without treatment, and the gray dots and the green dots are the treated wounds. And our prediction shows that the treated wounds should close faster than the control wounds, as expected. Now, this early study, we don't have a lot of statistics. We have some statistics, and what it shows is that when we do histology on the wounds at the end, we see that the epithelial thickness in the treated wounds is higher than the control wounds. The cradulation area is lower. We also have and the epithelial thickness is statistically significant, if I remember correctly. We also have some preliminary data on the various factors where some of the biomarkers that are related to inflammation tend to be lower in the treated wounds, and the biomarkers related to a wound that is healing appropriately tend to be higher in the treated wound. Of course, this is require more work. Additional data we found is that we have very nice growth of nerve fibers in the treated wound. This is statistically significant compared to control wound. And we also have lower collagen to 3:1 ratio, meaning that the wound is more maturated, but these results are trending because of the large error bar in the control wound itself. So fundamentally, this very large team of people developed a device that is able to do closed-loop control of wounds. And the idea is that basically it can augment the reach of physicians by monitoring the wound, deciding what the state of the wound and whether it requires treatment, and having an onboard automatic treatment that can be delivered bioelectronically. And this is the team, and also we had a lot of alumni. This is a long 5-year work, and now they all are pretty amazing places. And with that, I would like to thank you for your attention and thank my group, the current group, and funding from DARPA and ARPA-H, and I'm happy to answer any questions.
Thank you so much, Dr. Rolandi. Any questions?
I think you're muted over there.
Can people online hear me? I think I'm. Oh yeah, so they are. I'm unmuted here.
Any questions?
Hi. Hi, I have a question. So you used neural networks to make decisions. Why not use a parametric model? I mean, it seems like you can understand all the, the interplay between the inputs and the outputs. Why not just use a parametric model?
So that is a better question for Professor Gomez, who's the person that is in charge of the control. The short answer is that we do not understand the correlation between all the entry points. And so like the goal here was to try to gather as much predictive power with as little data as possible to minimize the number of sensors on the wound. And so the advantage of using a neural network is that you can treat the wound as a black box. And basically, you don't necessarily need to understand what's going on, but all you need to do is to correlate the data from the sensors with the outcome. But if this is not a satisfactory answer, I would have to, you can email me and I'm happy to relay to Professor Gomez because she will have a much better answer than mine should.
No, that makes sense.